Hi everyone. In this video, we're going to take a look at the structure of viruses and an overview of how they replicate. Before we get into what they look like, let's take a moment to talk about what viruses are. Viruses are neither prokaryotes nor eukaryotes, and in fact aren't living organisms at all. A virus is essentially a container assembled from protein in which some genetic information is stored. The genetic information can take the form of DNA or RNA, and either can exist as a single or double-stranded molecule. When it comes to shape and structure, there's no simple one-size-fits-all with viruses. They can vary from relatively simple helical structures to icosahedral shapes and even these crazy spaceship-looking ones like this T4 bacteriophage here. In addition to the genetic material, is the idea that they all contain a protein capsid made up of individual protein subunits called capsomeres. There may or may not be additional structures, such as the glycoproteins and membrane envelope that you can see here on this HIV virus, for example. Now, regardless of structure, all viruses are what we call obligate parasites. Since they're not cells, they don't have the capability to perform any metabolic functions or to reproduce on their own. They can only do so inside of a host cell. So let's take a look at a basic overview of a viral life cycle. It begins when the virus attaches to a host cell through an interaction between a protein on the surface of the virus and a receptor on the host cell. This interaction is one of the key factors in determining the host range, the specific range of cells that a single virus can infect. Once the virus is attached, the cell will endocytose it, bringing it into the cytoplasm, where uncoating will release copies of the viral genome and any other components into the cell. This is where the virus takes over the host, effectively hijacking the cell's enzymes, amino acids, nucleotides, and any other molecules that it needs to replicate, transcribe, and translate the viral genome. Once the pieces are made, the new viruses will self-assemble, finally leaving the cell, either through exocytosis, budding, or cell lysis, depending on the type of virus and host. These progeny viruses can then go on to infect new cells, repeating the cycle in a new host. Thanks for watching, and please be sure to check out the videos on bacteriophages and eukaryotic viruses.